Welcome to the Sans Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. We really appreciate you joining us. Please remember to take just a second to like, subscribe, and share this deep dive with your friends and family. It genuinely helps support the Sans Science Experience. So, today we are venturing deep inside the cell. We're going to explore an organelle that, well, might just be the key to understanding how and why we age. We're talking about the lysosome.
Right. Most people think of the lysosome as, the cell's stomach or maybe its recycling bin.
Yeah.
It does do that, breaks down waste, old cell parts. But it's actually much more central. It's a master regulator. It coordinates metabolism, controls those really major lifespan pathways, like mTOR.
And there's been this growing idea for years, decades really, that when the lysosome starts to fail, that's a huge driver of age-related problems, especially nasty things like Alzheimer's and Parkinson's. Makes sense. If the recycling system breaks down, garbage piles up, right? The cell eventually just fails.
Exactly. But here's the big challenge. The lysosome is incredibly small, tiny. It's only a minuscule fraction of the whole cell's volume. So when scientists typically look at a whole tissue, any subtle, really important molecular changes happening inside that tiny lysosome, they just get completely lost, drowned out by everything else.
Right. It's like trying to hear a whisper in a crowded room. So the goal of the work we're looking at today was really ambitious then. They wanted to get past that limitation. To figure out, with real molecular detail, what exactly goes wrong inside the lysosome as we age, trying to find that specific signature of failure.
And that's where the clever methodology comes in. To see what's wrong inside, you first have to get inside cleanly. You need pure lysosomes.
Yeah, how on earth do you pull out just those tiny, fragile lysosomes from a whole chunk of, say, aged brain tissue without smashing them or getting junk from the rest of the cell?
They used a really neat technique, organellar metabolomics. Specifically, they used something called lysotag mice. Think of it like this. Imagine every recycling bin in a city suddenly had a unique magnetic handle bolted onto it. These mice are engineered so their lysosomes have a specific protein tag, TMM192, on the outside. That's the handle.
Ah, okay. So they can basically take a tissue sample, grind it up, and then use an antibody, like a magnet attracted to that handle, to just pull out the lysosomes, leaving all the other cell stuff behind.
That's pretty much it. It allows for super fast, very high purity isolation, which is critical because lysosome contents can change really quickly. And importantly, they also check their findings using a different method, a tag-free one, using an antibody against P18, just to be sure the results weren't some weird artifact of the lysotag system itself.
Clever. So armed with this tool, they created this lysosomal metabulum mirus senis, an atlas of what's inside the lysosome in different aged mouse tissues. What did this unprecedented peek inside reveal?
It showed something immediately. A really clear metabolic signature. And it wasn't random. It was consistent across 4 really key tissues: the brain, the heart, skeletal muscle, and also white adipose tissue.
A systemic change then, not just one tissue behaving oddly. Okay, let's get to it. What was the signature? What builds up in old lysosomes?
2 specific types of molecules accumulated to really high levels in aged lysosomes in all 4 tissues: the amino acid cystine, and these lipid precursors called Glycerophosphodiesters, or GPDs for short.
Cystine and GPDs. Those names ring a bell, but not in a good way. They sound familiar from disease contexts, right? This is where it gets fascinating, I think.
This is absolutely the core aha moment of the whole study, yes. Both GPDs and cystine are metabolites directly, causally linked to some really devastating severe lysosomal storage disorders, LSDs, that affect children.
Wait, so you're saying normal aging starts to look like these catastrophic, rare genetic diseases of childhood? Can you connect those dots for us?
Okay, let's take GPDs first. These build up massively in Batten disease. That's a fatal neurological LSD, often caused by defects in a gene called CLN3. Now, cystine. Its accumulation is the defining feature of cystinosis. That's another severe disorder where a faulty cystine transporter, cystinosin, causes cystine crystals to build up basically everywhere, leading to kidney failure and other problems.
So in those diseases, a specific genetic error causes a rapid early pileup of these molecules, leading to terrible outcomes. And this new research suggests that in normal aging, our lysosomes slowly start doing kind of the same thing, a low-grade chronic version.
That is precisely the implication. Finding these two specific LSD-linked metabolites building up during healthy aging gives us the first direct molecular evidence connecting normal geriatric decline, the stuff that happens to everyone with these very specific, rare, and often fatal childhood diseases. It fundamentally reframes how we might think about aging.
It's a bit chilling, like we're all slowly developing a mild, chronic form of these storage disorders over decades. But okay, for this to be a really solid finding, it needs to be more than just a quirk of one type of engineered mouse, right? How sure are they this is a general thing in mammals?
They were very thorough on that point. They confirmed the GPD and cystine buildup not just in the lysotag mice, but also in standard wild-type mice, using that tag-free isolation method I mentioned. And crucially, for relevance to us, they found the exact same signature, high cystine, high GPDs, in aged rats.
And rats are often considered a better model for human metabolism than mice, aren't they?
Generally, yes. They share some metabolic similarities, so finding it across species using different methods, it really cements this as a conserved molecular feature of mammalian aging, and that leads into why they call it a clock.
Yeah.
It's not just something that appears when you're old.
Right, the clock aspect. How precisely does this buildup track with age? Is it just, high in old animals, low in young ones, or is it more predictable?
It's remarkably predictable. They measured the levels across the mouse lifespan, young adult, middle-aged, old, very old. And they found a really clear pattern. Cystine and GPD levels don't just suddenly jump up late in life. They start increasing quite early, and they rise linearly with age. It's a steady, progressive accumulation.
Linearly. That implies you could potentially predict age based on these levels. How good is that correlation compared to other ways we measure biological age?
The precision was actually quite impressive. They reported high R-squared values, the statistical measure of correlation. These values were comparable to sophisticated aging clocks that people are developing, like ones based on spatial transcriptomics. So this linear predictable increase is what solidifies the idea that this isn't just a symptom, it's a quantifiable organelle-based biological clock, measuring age right at the level of the cell's waste disposal.
Let's zoom in on the brain for a minute, because that's where age-related decline is often most feared. The brain's complex, lots of different cell types. Did all brain cells show this lysosomal aging signature equally?
That's a really important point. No, it wasn't uniform across the board. They found the buildup of GPDs and cystine was most dramatic, really concentrated, in two specific cell types, microglia and neurons.
Microglia, the brain's immune cells and neurons, the main signaling cells.
Exactly. And critically, these are the very cell types most implicated in neurodegenerative diseases like Alzheimer's. Interestingly, other brain cells like astrocytes or oligodendrocytes, they showed much smaller, almost minimal changes in their lysosomal metabolites with age.
That cellular specificity is telling. It points to where the vulnerability really lies within the aging brain. Now, you mentioned small molecules, cystine and GPDs. Were there other changes? What about the lysosome structure itself, the lipids in its membrane?
Yes, they looked at that too. And alongside those metabolites, aged lysosomes in the brain, heart, and muscle also showed an increase in a specific type of lipid called bis-monoacylglycerophosphates, or BMPs.
BMPs, those are unique to lysosomes, aren't they? Part of their membrane structure.
They are indeed the signature lipid of the lysosome membrane, crucial for its function, like helping digestive enzymes work properly. But, just like the metabolites, when BMP levels get too high, that's also linked to certain LSDs, like Niemann-Pick disease. But here's the really interesting twist. The specific combination they found in aging elevated BMPs plus elevated GPDs plus elevated cystine, that particular trio isn't characteristic of any single known LSD.
So aging isn't just mimicking one specific childhood disease. It's creating its own unique kind of composite molecular problem in the lysosome, a triple whammy.
You could put it that way. It's like a bespoke pathology crafted by time itself. This combination BMPs potentially altering the membrane structure and function, while GPDs and cystine perhaps overwhelm the breakdown capacity, seems to define the specific molecular compromise of the aged lysosome.
Okay, so we have this clear, conserved linear clock ticking away inside our lysosomes. The obvious next question is, can we reset it? Can we turn it back? They tested this using caloric restriction, right? The classic anti-aging intervention.
Exactly. They put the lysosomal clock to the test using CR, which is probably the most robust, well-studied way to slow aging and extend lifespan in mammals. But how it works at the subcellular level, well, that's still being figured out. And the results they got were fascinating, but also starkly different depending on the tissue.
Okay, let's hear the good news first. Where did caloric restriction manage to rewind the lysosomal clock?
In the heart. The results there were clear and positive. CR completely restored the levels of lysosomal cystine and GPDs back down to what you'd see in young mice. It essentially rejuvenated the heart lysosomes, at least by this metabolic measure. It showed this clock can be reversed, which is hugely encouraging.
Fantastic news for heart health span. But I sense a but coming. What happened when they looked at the brain?
That's where the story takes a turn. In the aged brain, caloric restriction had essentially no detectable effect on the buildup of cystine and GPDs. Despite the whole animal being under CR, the lysosomal clock inside the brain cells just kept ticking upwards.
Wow. That suggests brain aging might be, I don't know, insulated somehow. Or driven by factors that systemic diet changes just can't reach effectively.
It certainly reveals really profound differences in how tissues respond at this deep subcellular level. And it highlights why this lysosomal clock is so valuable, right? It acts as a readout. It can tell you if an intervention is actually working where it matters most. If you're designing an anti-aging drug, this tells you getting effects in the brain might be a whole lot harder than in the heart or muscle.
Okay, so we've got the fingerprint of lysosomal aging cystine, GPDs, BMPs. We know it ticks predictably like a clock. We know it's reversible in some places like the heart, but stubbornly resistant in the brain, at least to CR. Which brings us to the really big final question the researchers themselves raised. Are these molecules, cystine, GPDs, BMPs, just inert junk piling up like a sludge in the bottom of the recycling bin because the machinery is slow?
Or, and this is the critical question for future therapies, are they actually doing harm? Do they become actively toxic, contributing to cell damage and decline, much like they clearly do in those severe childhood LSDs?
Because in Batten disease or cystinosis, that buildup isn't benign. It directly causes inflammation, cell dysfunction, cell death. If that same principle applies even slowly during normal aging,
Then figuring out how they cause toxicity becomes paramount. Understanding those mechanisms is the key. If we can figure that out, maybe we can develop ways to specifically target the production, breakdown, or removal of just these molecules, GPDs, cystine, maybe BMPs. Even if we can't stop aging overall, perhaps we could lessen the toxic load and keep critical organs healthier for longer.
this research really feels like it's provided a crucial piece of the puzzle, maybe even a new roadmap. Establishing this first organelle-based clock, showing the lysosome isn't just a passive victim of aging, but likely an active participant, maybe even a driver.
It's a powerful demonstration. The deeper we look inside the cell's workings, the closer we might get to finding real ways to influence not just how long we live, but, how well we live during that time.
Absolutely. And that seems like a perfect place to wrap up our exploration of the lysosomal clock of aging. Thank you for joining us on the SANS Science Experience. If you found this deep dive into the molecular mechanics of aging interesting, please do remember to like and subscribe for more insights into the latest science. We look forward to having you with us next time on the SANS Science Experience.